Hi, today we're going to talk about the scientific process. People often have the wrong impression about biology or any science. They think that because it contains a bunch of facts and ideas about the natural world, it will never change. However, science is actually always changing. In fact, a lot of the biology that we will learn this year may change and be revised in the future with the discovery of new information. Science is an organized way of gathering information about the natural world, not supernatural phenomena like ghosts and spirits. After scientists spend time looking for patterns and connections, they propose explanations based on the evidence, not based on what they believe. This process is continuous as scientists are always in search of more information. One goal of science is to provide natural explanations for things of the natural world. It also uses those explanations to understand patterns in nature and then use them to make predictions about other natural events. Usually, it is more about what we don't know than what we do know. The process of science is something that you will use all the time, even though you may not be aware of it. You use it to solve everyday problems like why your car won't start. The scientific approach is pretty much the same way. Overall, a lot of people oversimplify the scientific process, and they put it into too few arbitrary Betty Crocker recipes instead. It's a much more complex, but we are going to have an order to it regardless. But you're going to learn that the order does not have to go in precise 1, 2, 3 fashion. On your paper, there's this graphic of the steps of the scientific method, and we're going to go around and talk about each one of them. Step number 1 is observe and define the problem. So in step 1, scientific investigations always begin with an observation. They have to. Although scientists gather this information by using their senses, it involves more than just looking at stuff. A good scientist will observe something that leads to a question nobody has really asked before. The second step of the scientific method is to research the problem. Just to make sure that nobody has answered the questions before, scientists must spend a lot of time finding out what is already known, or more importantly, not known by doing research. Most research is done by going through scientific magazines called journals where scientists publish their findings. It could be quite elaborate and painstakingly long and to actually collect all this research on that problem. Step number 3 is to then make your hypothesis. After observing and researching, scientists will make inferences to help formulate a hypothesis. An inference is an interpretation or conclusion based on what you already know or can observe. For example, if you look down here at the die, if it's showing the 5 and the 1 and the 3 and the 6 and the 4, what would you say is on the bottom? Well, by looking at the available evidence, you could infer that the 2 would be at the bottom without actually ever seeing it. This is what an inference is. And scientists use inferences to make hypotheses. You probably learned a hypothesis is an educated guess or a best guess. Although that's not entirely correct and we need to develop your understanding of the scientific method, a better definition would be, and this is the one you need to know, is that a hypothesis is a proposed explanation for natural events that can be tested. Hypotheses must be testable. That's the key. They have to be testable. In fact, most hypotheses are made in statements called if-then statements that show the possible relationships between two things and then tells what your prediction is. For example, if leaf color is related to temperature, then exposing plants to cooler temperatures will result in changes in leaf color. The next step of the scientific method is to design an experiment and conduct it. It has to be a controlled experiment. And so in a controlled experiment, that's the key, it means that you should only change one variable, while everything else is kept constant, which means the same. If you change more than one variable, then it makes it very difficult, yet impossible, to tell what's the cause for the result that you got. For an example, let's say that if you want to know if fertilizer helps your tomato plants grow, you would need to set up an experiment where some plants get fertilizer and some don't. But if you also water one plant more than the other, and then another plant gets more sunlight, in the end, what caused the change? The fertilizer, or was it water, or was it the sunlight? You're not going to know. That's why it's important to only test one thing, one variable. The variable that is purposely changed by the scientist is called a manipulated or independent variable. In the experiment with the tomato plant, the fertilizer would be the manipulated variable. The variable that is observed to change in response to what the scientist manipulated is called the responding or the dependent variable, because it is dependent upon what you change. In the tomato plant experiment, the responding variable would be the height or growth of the plant. When you graph this data, the manipulated variable always goes on the x-axis and the responding variable goes on the y-axis. So down in this graph over here, the number of bubbles produced per minute by plant X in various depths of water, the manipulated variable that you could control would be the depth of water, so it goes on the x-axis down here. But over here, the y-axis is the responding variable, which is the amount of bubbles produced per minute. Most experiments are divided into control groups and experimental groups. A control group is exposed to the same conditions as the experimental group, except for the manipulated variable. Anything that is kept the same between the two groups are called controls. There are usually many. For example, in the tomato plant experiment, controls would include the amount of soil the plants got, the type of soil, the size and the type of the pot that they're planted in, the amount of sunlight, the temperature, the amount of water. They all have to stay the same. Controls are important because they allow you to analyze your results more effectively since you have something to compare to. The next step of the scientific method is to organize and analyze data. Scientists need to make detailed records of data in their experiments. There are two types of data that you need to know that need to be recorded. Quantitative is any numerical-based data, numbers. Heights of the tomato plant would be an example. Qualitative are descriptive observations that cannot be counted, such as color, shape, movement, behaviors of certain animals, etc. Both kinds of data are very important and therefore must be taken very accurately. The next step of the scientific method is to make a conclusion. Once the data is organized and analyzed, scientists will use it to support or reject or revise their original hypothesis in order to make a valid conclusion. It is important to know that this cannot happen by doing an experiment just once. Most often, the data collected will force the scientists to reevaluate their hypothesis or experiment and then redo the whole thing to get better data. This usually happens many, many times before an actual final conclusion can be made. When the conclusion is made, it is made free of bias. And what that means is that it's free from feelings or beliefs of that scientist. It is also important to note that just because a scientist may not particularly like the data that he got, he can't just dismiss it like it never happened. If the company that he works for finds out that he did this, he could be legally prosecuted for falsifying data. The last step of the scientific method is to communicate results. Communication and sharing ideas is essential to the scientific process. Publishing allows scientists to reevaluate each other's work. Scientific articles are published in science magazines called journals. There are hundreds of different scientific journals for all the different disciplines in science. Two very famous scientific journals are Science and Nature. These articles are pretty much a very detailed lab report that includes introduction, materials, experiment, controls, data, analysis, and conclusion. During the peer review process, scientists will share their findings by publishing articles that have first undergone peer review by an anonymous expert, or more than one. The reviewers will provide feedback to the journal editors, and the editors will send those comments back to the original scientist to be revised before it could be submitted again to be published. This process can occur several times before the research is published for all to see. Once research has been published, it often sparks new questions in the scientific community and can be used by other researchers to base their own experiments on. And those are the steps of the scientific method. Now, one thing I hope you noticed with this graphic is that it kind of goes in a circle. You always have observe and define the problem, and then you have the other steps that kind of go around it, but it never points back to this. I want you to realize that once you have a scientific problem, you research it, you make the hypothesis, you design and conduct the experiment, but it doesn't have to go in this order. So let's say you have the problem and you research a problem and you find that it's already answered. Well, then you're done. Let's say that you research the problem and you want to find more about it and do an experiment and make a hypothesis. And then to make the hypothesis, sometimes you have to research more, so you'll come back here to number 2. And then you made a hypothesis, and then you need to design an experiment, but you don't know where to start, so you have to go back and research what other people did. And then you could do your experiment, but maybe you have to redo your hypothesis, but eventually you get your experiment done and you get the data, but your data stinks, and so you have to go back and research more and come back and design a new experiment and then get some more data, and then you have to have more data, so you do the experiment again, you get more data. Eventually you can make a conclusion whether or not you like it. And so this is an ongoing process that is very messy and doesn't necessarily have to go in these steps, but in general it does proceed in a kind of a order such as this. The last thing I want to talk about are theories. Oftentimes, the scientific term theory is confused with an everyday theory. An everyday theory is just a hunch or a guess about something with much uncertainty. However, a scientific theory is a scientific idea that has been well tested, well researched, and well supported by the scientific community. Evidence from many studies may support many related hypotheses. In this case, theories are formed to tie them together. So, when scientists talk about gravitational theory, evolutionary theory, atomic theory, they're referring to much more than some sort of guess or hunch. Over time, these theories may become the dominant view among the majority of scientists. But no theory is considered absolute truth. Remember, science is always changing as new evidence is uncovered. A theory may be revised, but rarely it is overturned. And I hope that was helpful.